Cells are like tiny factories, constantly bustling with activity. To keep the lights on, they need a steady supply of energy. This energy comes from the food we eat, primarily glucose, a simple sugar. Our cells break down glucose in a process called cellular respiration. The initial steps of this breakdown occur in the cytoplasm, the jelly-like substance that fills the cell. Glycolysis, meaning sugar splitting, is the first stage in this energy extraction process. During glycolysis, one molecule of glucose is broken down into two molecules of pyruvate. This process generates a small amount of ATP, the cell's energy currency. Imagine ATP as small rechargeable batteries that power various cellular processes. While glycolysis provides some ATP, it's just the tip of the iceberg. The real energy payoff comes from completely oxidizing pyruvate in the mitochondria, the cell's powerhouse. Mitochondria are like tiny power plants within our cells, responsible for generating the bulk of the ATP required for cellular functions. The transition from glycolysis to the next stage of energy production takes place as pyruvate molecules journey from the cytoplasm into the mitochondria. This journey marks the beginning of a much larger energy-yielding process. Pyruvate molecules produced from the initial breakdown of glucose cannot directly enter the next stage of energy production. They need a special pass to get into the mitochondria. This pass comes in the form of a helper molecule called coenzyme A, or CoA for short. Inside the mitochondria, pyruvate undergoes a crucial transformation. It loses a carbon dioxide molecule and links up with CoA, forming a new molecule called acetyl CoA. This conversion is like changing your clothes to fit in with a new crowd. Acetyl CoA is the key to unlocking the full energy potential of glucose within the mitochondria. The formation of acetyl CoA marks the transition from glycolysis to the next stage of cellular respiration, the citric acid cycle. This cycle, named after its starting molecule citrate, is a metabolic merry-go-round that extracts more energy from the remnants of the original glucose molecule. Acetyl CoA acts as the fuel for this cycle, driving the production of more ATP. Once inside the mitochondria, acetyl CoA enters the citric acid cycle, also known as the Krebs cycle. This cycle is a series of chemical reactions that resemble a merry-go-round, with molecules hopping on and off. Acetyl CoA combines with a 4-carbon molecule called oxaloacetate, forming citrate, the cycle's namesake. As the cycle churns, citrate undergoes a series of transformations, losing carbon dioxide molecules along the way. These lost carbons are exhaled as waste products of cellular respiration. The cycle's primary goal is not to dismantle citrate, but to generate electron carriers, molecules that shuttle electrons to the next stage of energy production. Two crucial electron carriers, NADH and FADH₂, are produced during the citric acid cycle. These molecules act like shuttles, carrying high-energy electrons extracted from the breakdown of glucose. These electrons are vital for the final stage of cellular respiration, where the bulk of ATP is generated. The electron transport chain, embedded within the mitochondrial membrane, is the final and most productive stage of cellular respiration. Think of it as a cellular relay race, with NADH and FADH₂ passing electrons from one molecule to the next. As electrons move down the chain, they release energy, like a ball rolling down a hill. This released energy is used to pump protons, positively charged particles, across the mitochondrial membrane, creating an electrochemical gradient. This gradient acts like a dam, storing potential energy. The flow of protons back across the membrane through a special enzyme called ATP synthase drives the production of ATP, much like water flowing through a turbine generates electricity. This process, called oxidative phosphorylation, is responsible for generating the majority of ATP in our cells.